Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Artur Stanchak, Application Specialist, Process Chromatography from Bio-Rad Laboratories.
Thank you for the introduction. I look forward to discussing today the monoclonal antibody purification process development using mixed-mode resins in the DOE approach. In the first part of my talk, I'm going to introduce you to quality by design concept, where it actually came from, why it's important, and how it's linked to the design of experiment. I will show you benefits of implementing DOE approach in your experimental work. I will also briefly discuss various mAb purification workflows that are used in biopharma production. In the next part of my presentation, I will show you two examples of the mAb purification using mixed-mode resins and the importance of implementing DOE approach to speed up your process development and maximize the process performance. Regulatory authorities such as FDA or EMEA published guidelines for pharmaceutical process development. The concept called quality by design became a successor for a legacy approach called quality by QC or quality after design. The old concept, in brief, postulated controlling quality of the product by testing it using a range of analytical techniques. Modern approach, however, in addition to quality control, also includes systematic approach to development, which begins with predefined objectives. The emphasis is put on deep understanding of the product and process in a risk-based approach. The design of experiment is then an experimental method to achieve quality by design. You will see later how DOE can improve your knowledge about the purification process, quality attributes behavior, and potential interactions between tested factors. The traditional approach to experimentation is called OFAT, which stands for one factor at a time. In short words, it implies changing one factor, for example pH, while holding all other variables constant. It is a straightforward approach, but it's very inefficient. On the response surface plot, you can see the mAb recovery on mixed-mode resin, Nuvia A Prime. Black dots represents, as an example, the test points for pH and conductivity in one factor at time approach. In this case, 9 chromatography runs failed to discover the optimal conditions for this molecule, which are marked in yellow. In addition, potential interactions between pH and conductivity cannot be detected. The design of experiment approach is very different as it studies multiple factors changing at once. As an example, the same response surface plot is presented as on the previous slide. However, this time dots are distributed in a completely different way. We can see that they cover much larger testing area. But it's worth noting that the ranges of pH and conductivity were the same. The key is just different way of combining them in experimental procedure. In this case, by leveraging the DOE approach, we were able to find optimal operation windows with only 7 chromatography runs. We are also able to discover interactions between factors. So the design of experiments is defined as a branch of applied statistics that helps with planning and interpreting experiments. It is a software-intense, systematic approach to investigation of the process. It is important to mention at this point that you will have to use software of your choice to help you with designing and processing the data. The simplified DOE process scheme includes multiple input factors that can be manipulated. Processing of biologics involves complex mechanisms. Each processing unit has a unique noise factors, biases, and level of precision. Therefore, it's important to include at least 3 center points to assess process variability. Also, a high variability of analytical methods might play an important role in assessing the DOE data. This brings us to the output of the process, so-called responses. I've mentioned already some examples of factors that can be manipulated to measure the effect of the desired response, such as pH and conductivity, which are examples of continuous type factors. Other worth mentioning are loading density, volumes of buffers used, residence time, or gradient slope, but there could be many more. The example of discrete factors is, for example, buffer type. Responses can be divided on two groups. One is a product quality attribute, and this includes purity, aggregate, and impurities levels, charge variance profile, or any relevant quality attribute. The second group includes process performance indicators, and this includes dynamic binding capacity, protein concentration, and others. Purification of MAbs is a well-established process. The majority of projects have a three-step purification workflows with protein A resin as a capture step. It gives excellent impurities clearance, but the process is lengthy and expensive. Alternative to that could be a three-step purification process but without protein A resin. It's more cost-effective, rich protein A ligand is not a concern anymore, and it provides a good impurities clearance. However, the development of a capture step might not be intuitive. Also, process yield can be negatively impacted. It's worth noting that different mixed mode resins can be placed in actually any stage of purification process. Please note that these workflows showing only chromatography steps, and for simplicity, other downstream processing steps like a virus filtration are not included. There is also another alternative to a standard three-step purification process for mAb. It's a two-step purification workflow with protein A coupled with mixed mode resins. This setup gets out the most from a mixed mode resins. There are several advantages for a two-step process. It's a cost-effective, short and simplified. It gives an effective impurities clearance with a decent product recovery. In the next slides, I will show you two examples of mixed mode resins being used in a two-step purification workflows. The first one will be Nuvia A Prime 4A, a hydrophobic anion exchanger used in a flow-through mode. The second will be ceramic hydroxyapatite media. It's a cation exchanger with a metal affinity used in the bind and elute mode. These are two orthogonal resins with a different modalities, used in two different modes of action. On the screen you can see a chromatogram from Nuvia A Prime 4A run, which was done in a flow-through mode with conditions optimized specifically for the reference Herceptin project. Very similar conditions were used for the purification of three different MAbs. Chromatograms, however, are not comparable to the reference process, and you can see a distinct tailing observed in each case. UV traces for mAb with the same average isoelectric point, like mAb 2 and mAb 3, are also showing differences. This suggests that the PI value is not the only one parameter that is important to predict the purification behavior. Other parameters, like distribution of hydrophobic patches on the protein surface, might also play an important role. If we look on analytical results, mAb 1 and mAb 3, despite the differences in UV traces, showed similar results to the reference with a very good aggregate and HCP removal. mAb 2, on the other hand, at the same generic conditions, show not as promising results. Regardless of mAb tested, the HCP level was very low. Analyzing chromatograms and analytical data, we acknowledge the fact the platform conditions are not applicable for every map and should be further optimized. This is why we decided to perform a DOE study to establish an optimal conditions for each map. We use for that purpose a DOE design called Phase Central Composite with total number of 11 runs, including 3 center points. The study was done on Foresight 96 well plates filled with 20 microliters of Nuvia A Prime resin. Factors tested were pH and conductivity. pH in the range between 4 and 8 and conductivity in the range between 5 and 45 millisiemens per centimeter. Here are contour plots for the yield response for Nuvia A Prime 4A. The red color shows higher value, blue indicates low value. We can see that the provisional optimal operational windows, yellow boxes, cover different ranges for different maps. Looking at these contour plots, it is easier now to understand why the UV traces you saw on the previous slide show so different profiles depending on the map used. It is clear indication that to save time and resources, the design space should be established even before trying generic conditions, which worked previously for other molecules. Here you can see a chromatogram from CHT media run, which was performed in the bind and elute mode under conditions optimized for the reference Herceptin project. Very similar conditions as for the reference process were tested again for 3 maps. Chromatograms are similar with only some differences in the retention time. In order to get more information about the purification performance of these 3 maps, a DOE study was executed on CHT resin. It was a full factorial design performed using 1 ml column and NGC chromatography system. It was a very simple design which required only 4 runs. Factors tested were pH and the linear flow rate. These are response surface plots generated by DOE software. I would like to point that for all 3 maps the responses showed very similar patterns. It's worth noting that numbers of scales are different. These results correspond to the entire peak eluted in the salt gradient. Without any collection criteria adjustment, what would happen in the next optimization round? Provisional operating windows marked as the yellow boxes, considering monomer content and product recovery. For different maps, the optimal windows have various ranges and are not in the same positions within the design space. This observation showing the need of performing the DOE studies in order to find optimal condition, even in the cases where all maps are showing similar chromatograms. On previous slides, I show you counterplots analyzing performance of Nuvia A Prime and CHT one by one, and the need for screening broad range of conditions using DOE approach to determine optimal operating window. Here, on this slide, I would like to show you the need of including orthogonal resins in your development work. This might help finding the optimal solution for your molecule much faster. Comparing the performance of orthogonal resins, you will find out quickly which of them provides better results without the need of redeveloping an existing platform process, which can be anyway suboptimal regardless of the optimization that has been done. For example, in case of map 1, similar purity and yield can be achieved using both resins. For map 2, better purity and yield can be achieved with CHT media, whereas for map 3, better purity and yield is obtained using Nuvia A Prime 4A resin. In summary, design of experiment approach is a very useful tool to improve efficiency of the process development. It can help you quicker and more deeply understand the process that can particularly be useful when working with a mixed mode resins. We can see on the field that two-step purification workflows with mixed mode resins become more and more popular. Resins such as present at Nuvia A Prime and ceramic hydroxyapatite are proven to eliminate the product and process-related impurities, including viruses. It is important to screen virus mixed-mode resins for every project. By testing orthogonal resins, the optimal solution can be established in a shorter time. Also, if they can operate in a different modes like these two examples, it can give you more flexibility in the designing of a purification process. Thank you for your attention. I would be happy to answer any questions at this time.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, how many parameters or factors can I include in my DOE study?
Yeah, that's a very practical question, but the answer is quite complex. So theoretically, there is no limit for numbers of factors that can be tested in the DOE experiment. However, to get out the most from the DOE approach, you should find suitable for yourself the balance between numbers of factors to be tested and the resolution to be achieved. So for high numbers of factors, typically above 9, the main objective is to find those showing significant impact on tested responses. It's called a screening design. Factors with non-significant effects can be then eliminated from the next round of testing. The main objective for the next, more in-depth testing is to find interactions between factors. This is known as a modeling design, and you will be looking to test between 4 and 9 factors there. The most information about the process interactions can be obtained by using so-called an optimization design, but it is not recommended to test more than 4 factors in this setup.
What are the typical factors to be tested for Nuvia A Prime 4A and CHT?
Yeah, that's a very good question. So, firstly, be aware that this might be a very product specific, and even the same molecule type might have a different range of significant factors. It is important to note that the factors are different depending on bind and dilute or flow-through mode is used. So for Nuvia A Prime, significant factors might be pH and conductivity, but also a buffer type. So residence time, loading range, and collection criteria normally also play an important role. In case of CHT media used in the bind and elute mode, the pH and conductivity of the load, but also elution are important. In this case, also residence time, but and also gradient slope, loading density, and UV collection criteria are the factors that should be evaluated. So these are the typical factors worth considering, but there could be others that influence your process. So this should be evaluated later on during your process characterization studies.
And our next question is, has a mixed mode resin replaced an IEX and Higgs steps in industry? Is there viral clearance data?
Oh, thank you for this question. Yes, we can see that the mixed mode resin, generally speaking, are gaining more and more interest in the field. There is a known example in the industry using a Nuvia A Prime resin, which is a slight hydrophobic anion exchanger. Obviously, the level of HTP and DNA was within the spec. It is a nice example as the virus clearance in this project reached a decent value above 5 log reduction, and that was for the xenotropic murine leukemia virus. Remember that the effectiveness of the virus clearance is a very product specific and it depends on many factors. But if you want to have an idea in what direction to go in your process development, please email us, so we'll be happy to share with you the application note regarding viral clearance, which is also available on our webpage. In this application note, we present a DOE study where we characterize the design space regarding virus removal. So you will be looking to learn from there which factors are important to control and how strong is their effect on the virus clearance. Also, you can find there the actual counter plot, which is, in my opinion, a good visualization, and it's nice to see what conditions are optimal with regards to virus clearance.
All right, well, it looks like we're running short on time, so if we don't get to your question, we'll be passing them along to Artur, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what DOE design should I choose in the first instance?
Yeah, it's a very good question. So, at the beginning of the process development, the most important is to test the broad range of conditions. So, select relevant factors such as pH, conductivity, product loading, and other I mentioned before, based on your prior knowledge and the risk assessment. So, selecting appropriate factors for test is more important than selection of the best DOE design at this stage. So for example, if you select the test and control only pH and forget about conductivity, it might turn out that the results will be misleading because there is no visibility of how conductivity will influence the process. As I mentioned before, selection of the DO design is driven by number of factors to be tested, but also important is to know how many chromatography runs you can afford. So knowing these two factors, you can put them in the software, and this will provide you with the best design matching your requirements.
Okay, well, thank you, Artur.
Okay, thank you for joining us today.
And thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.